Okay, good afternoon, everyone. Thanks for being here with us today. Greeting from Navasayadri Institute of Pharmacy. It gives us immense pleasure to announce that on occasion of World Pharmacist Day, 25th September 2021, there will be a short webinar from Professor Joanna Bridger about the Progeria research. We will also announce poster winner and continue with MOU signing with Genome Engineering and Maintenance Network. So now I request Dr. Praveen Bade sir, please give a brief intro about Professor Joanna Bridger. Please, sir.
Mic name.
Hi.
Hi, Professor Joe. How
Oh,
are you?
very nice to see you, Pravin.
Nice to see you. So yes, I think just Madam just mentioned about the MOU, but it's not about MOU. It will be a collaboration. So research collaboration, we will say.
Okay.
Yeah. So just want to. It is my pleasure that I'm introducing Professor Joe. Uh, I had an opportunity to work with her about 3 years, 3 to 4 years, with Joe and Dr. Kill. So she has established her own independent laboratory in nuclear and genomic health in Brunel University. Her research concerned how the genome is spatially organized, influenced, and manipulated within its environment, the cell nucleus. The group has had a numbers of major advantages and is currently focused on aspect of genome behavior in replicative senses and the premature aging diseases such as progeria. Also working on host pathogen interaction and female cancers. So she want to understand how uh, structures such as nuclear lamina, nuclear skeleton, and nuclear motor influence the function of the genome. Uh, Professor Joe also teach cell biology at level 2 and 3 at Brunel University. Also, development of biology at level 2, gene expression and epigenetic and cellular organization in aging for master's level students. And yes, the lab is always full with PhD students and postdoc students, and I have experienced that. Uh, She's head of Genome Engineering and Maintenance Network established, which work in genome biology and has developed over time in biosciences at Brunel. And today we are on the occasion of World Pharmacy Day. We had a opportunity to have a talk from her on Progeria research. Um, so we are going to share the slides. So slides is not a problem. So I will now pass on to Professor Joe, and we'll start the presentation.
Thank you, Pravin, very much. It was a superb pleasure having you at Brunel. You were so driven, so dedicated. It's a privilege. So, and today is a privilege too. So thank you very much for that.
Thank you. Thanks.
Yes, there are my slides. That's good.
Yes.
Isn't this amazing? I mean, it's Saturday morning here, and we're talking across the continents. It's wonderful. Okay, so that's the first slide.
Yeah, can you can see the slides, yeah?
Yes. So do you want to do the showcase, the show? Should I present it like that, or do you want to push the button for the showing
No, I
them
think
full?
this will be fine.
Okay, fair enough. Okay, so yes, wonderful to talk to you today, and I want to talk to you about this premature aging syndrome called Hutchinson-Gilford Progeria syndrome. And it's something that my husband and I, and there's a picture of him here, Pravin will recognize him, Dr. Ian Kill. So we set out to work on this together a number of years ago because he was interested in aging and I was interested in the lamin proteins that we find inside the cell nucleus. And it so happened that I was at a meeting in America where they announced that the mutation that caused Hutchinson-Gilford Progeria syndrome was in lamin A. Was in the lamin A gene, and this was part of the cell nucleus. So we came together and decided we were going to work on it together. So that's where it all started. So next slide, please. And we're from Brunel University, which is in London. Sometimes people think it's in Bristol because of its coming from the engineer that was in Bristol. So is that slide coming up? Yeah. I can't see it. Okay, so I can't see the next slide. But what I can do is I can look at my slides here and just talk to them. Does that work?
Yeah, it's fine.
Okay, I'll do that. Right, so we're then at Brunel University, which is actually very near Heathrow Airport. It's only like a 10-minute drive. And so you can see on the slide Windsor Castle and then Buckingham Palace inside London. And so we're sort of somewhere between there. And there's a picture of the lecture centre there of Brunel. So if you're coming through Heathrow, it's not very far to come and visit us. So then on the next slide, I want to talk about Hutchinson-Gilford Progeria syndrome. So it's a very rare disease. And it's a very difficult disease for the families to handle because these children, they're born quite normal. They seem like they're going to flourish and be fine. And then after a little while, usually about 1 year old, they get, the parents start to notice that there are issues with growth and movement and things start to go wrong. And these children have dramatically foreshortened lives. So they only live into their middle teens. So they only sort of make it to teenage years. And they show a lot of signs of premature aging. So you can see here the little girl with the blue dress. She's lost her hair. And then this is Hayley Okines, who was a girl with progeria in the UK, and she came to visit us at Brunel several times, and this is one of her visits. And I think she's about 10 years old. Again, you can see that she looks a lot older. They die usually from heart attacks and strokes, like it sort of usually happens in normal aging, and it's, as I said already, it's due to one base pair change in the lamin A protein. And here in green, in the cell nucleus, so we have a normal cell nucleus at the top of that panel, which is nice and sort of round, ellipsoid if you like, and that's how a normal cell nucleus will look. But the one underneath, you can see that it has invaginations and herniations, and it looks really dramatically different. And that's a real telltale sign of a progeria cell, where you don't get this proper structure at the nuclear envelope. So that's the cell nucleus there. What happens with this mutation, this single base pair change, it changes a splice site. Okay, so it creates a new splice site that then leads to a really toxic protein, which is it is still a lamin A, but it retains a farnesyl group, and I'll go through that, the biochemistry of that in a minute. So people look for this misshapen nuclei, but we have some data to show that this is just the worst part of the sort of cell cycle for these, the cell lifespan for these cells, because they can be severely affected, the cells in the tissues, yet look normal by shape. So it's not a great measure, and people have been using it across all sorts of trials and research to use the shape change as a measure of a drug working, and we don't believe that is a very good way of doing it. So I'm going to move on to the next slide, which describes that this progerin, this is what we call this lamin A that has the mutation in it, we call it progerin after the disease, progeria. But this is why it's a toxic protein. So the normal lamin A processing that you can see in panel A, okay, we have the protein, the pre-lamin A it's called, made, translated, and a farnesyl group is added to that. The farnesyl group is thought to be important to get the lamin to the right place in the nucleus, which is the membrane, the inner nuclear membrane of the cell, and so therefore it's needed. But it then goes through this sort of complicated remodeling, if you like, where this farnesyl group is actually then cleaved. And so mature lamin A doesn't have that farnesyl group. It's the farnesyl group that makes these lamins, these toxic lamins, poisonous to the cells, because it then has that farnesyl group and association with the membrane when it shouldn't. Like when cells are going through mitosis, the lamin needs to come away from the membrane, but it can't in the progeria cells. So I think in the middle panel, panel B, you can see that even though we've got to the end of this process where we've got progerin, you still got that farnesyl group on the end. This is normal for the other type of lamins, which is lamin B1 and B2, but they're also then they need to be with the membrane, but then we get cleavage as well. So this is why this protein ends up affecting so many tissues in the children, but not the brain tissue, not nervous tissue, and it doesn't affect their immunology either. It's tissues that seem to be under some kind of stress. So if I move on to the next slide, that's what I'm doing now. You can see the mevalonate pathway. I'm not very good at saying that word. Anyway, but this is the pathway that leads to getting the farnesyl group. Okay, and people have looked at this pathway thinking, aha, if we can stop the farnesyl group being made, so there's an enzyme called a farnesyl transferase that puts the farnesyl group onto the protein. If we can interfere and interrupt this pathway with different drugs, then perhaps if you don't have a farnesyl group, then the protein won't be as toxic. And this is what people have done. So they've looked to farnesyl transferase inhibitors, but also statins and bisphosphonates also impact on this pathway. And so people have used those drugs in trials and suggested them. So we also wanted to look at those drugs to see what's happening in the progeria cells that we were interested in. So I'm going to the next slide now. So we were funded by a really nice funding body called Sparks, and they are interested in funding diseases of children, but also rare diseases, because it's quite difficult to get funding for rare diseases these days. You know, if it's only, I mean, there's probably only 600-800 children in the world at any one time that have progeria, so it is rare. And in a country, there may be, at the moment, I think we have 3 progeria children in the UK, and you will have a number in India, and there has been some documentaries made about progeria children in India before, but it's hard to find them all sometimes across the world. So this study was funded because we can look at these drugs, but also other drugs that people had suggested could be really interesting. So the farnesyl transferase inhibitors are there, but if you then block farnesylation, you can get then geranyl moieties put onto the protein. So we also wanted to block that as well, because that can substitute for the farnesylation. So you've got pravastatin, which is a statin, zoldronic acid, which is a bisphosphonate. But the French researchers wanted to put these two together. So then there were combinations of drugs that the children were taking, and now still are on another trial at the moment. So there was a combination of FTI, pravastatin, and zoldronic acid. Rapamycin was a drug that came in also to see if it could help with DNA damage, and I'll come on to that. They're also in these cells. There's a lot of DNA damage, which is really important to the discussions today. So, and other drugs like N-acetylcysteine and insulin-like growth factor were suggested in some papers. So we looked at all of those. So I'm going to the next slide now. And we wanted to look at the combinations that were actually the children were actually taking. So there were these drug trials at the time, FTI on its own, pravastatin and zoldronic acid only, FTI, pravastatin and zoldronic together. So it was really important that you look at these things in combinations. If they're going to be taken by a person, then what happens if you have mixtures of these drugs going into the children? And now the new trial is FTI and rapamycin together. So rapamycin has come out as an interesting drug, but it also causes quite some considerable side effects as well, and that has to be taken into consideration. So moving on to the next slide, just to describe a little bit about what I do, which is I'm very interested in, as Pravin said, the organization of the nucleus. And we have these chromosome territories, and you can see here in the bottom panels where we've painted individual chromosomes with specific probes that pick up chromosome 18 is there in green and chromosome 19 in red. And you can see that they're in specific areas, territories. They're not spread out all over the nucleus. And part of organizing these chromosomes is the nuclear lamina, where we find the laminae, and in these children we find progerin. So it has a big impact. We thought potentially has a big impact on chromosome behavior, which then would then affect gene expression. So we wanted to look at that. So we have been moving on to the next slide. So we have been looking at position of chromosomes inside the cell nucleus for a long time, because we was part of the group that discovered that not only were the chromosomes not sort of the DNA all over the place, but they were in these territories, but these territories have their actual own addresses, positions within the cell nucleus. So on this 2D and 3D analysis slide, hopefully you can see that there's in the corner, we outline a cell nucleus, which is in blue, which is a DNA stain, DAPI, and then we delineate the chromosomes, and there's 2 chromosomes in green. And that using this method, we're able to position the chromosomes. And so you can see this is the graphs, just so, you know, the best case scenario. These are cartoon graphs really of what it could look like if the chromosome was towards the periphery of the nucleus, at the edge of the nucleus, in an intermediate distribution, which is in the sort of between the edge and the middle, or in the middle, in the interior of the nucleus. And we can do this by 2 and 3D. We can also then look at proliferating cells. So cells are actively going to divide with a marker called Ki67, and then non-proliferating cells we see with no marker. And what's very interesting about the Progeria cells is that they are proliferating like crazy. So they have this marker, but they look like they're not proliferating when you look at their chromosome distribution. So that's a really fascinating thing to do with Progeria cells. So I'm going on to the next slide now, which is when we're looking at the Progeria fibroblasts, so these are the skin cells, we found that chromosomes were mislocalized. I know there's a lot of graphs on here, but I do want you to just focus in on, I can't use the pointer, but if you look at the top row, we've got across there the graphs for chromosome 10, chromosome 18, and chromosome X. And you can see that chromosome 10 is in an intermediate position, chromosome 18 is skewed towards the edge of the nucleus, and chromosome X is really up against the edge of the nucleus. And then if you go down to the bottom 3 rows, you can see that there's quite different, not for chromosome X because that's the control, but the chromosomes are in different locations in these 3 Progeria cell lines. So the take-home message from that is the chromosomes are not positioned correctly in the Hutchinson-Gilford Progeria cells. So I had a wonderful PhD student called Ishita Mehta, and she came from India and worked in the lab with me for a number of years. And she treated the Progeria cells with the FTIs, so the farnesyltransferase inhibitors. And she was able to rescue the chromosome position. So the chromosomes went back to their proper places. And I think you can see here across the top with chromosome 10. All right, so we've got untreated, the progeria cells, just the DMSO that the drug is delivered in. And then when you've got FTI and FTI plus GGTI, you get a change in position. Same, well, much more dramatic for chromosome 18, who is in the interior in the progeria cells and then moves to the interior, into the periphery when you treat with the drugs. So I'm going to now move on to the next slide. Okay, and then we also wanted to not just test the FTIs, but all the other drugs as well. And what came from this, and this was work done by Craig Clements and Mehmet Bickel, that we were only able to put chromosomes back to the right locations when FTI was present in the combinations. It didn't matter if you had other drugs in there, and the other drugs like the statins and the bisphosphonates, they didn't affect the chromosome position. So FTI was looking really good, and the children were on the FTIs and they were improving on the FTIs. So I'm going to the next slide, which is a cartoon of chromosomes whizzing across the nucleus. So this is something else we discovered, that if you stimulate a cell by taking away its food or giving it a heat shock, you can get very rapid active movements of individual specific chromosomes. Not all chromosomes move, so it's not a reorganization. So chromosomes can move very rapidly. And what we found was in the progeria cells, we didn't have this repositioning. We think this repositioning is really important for the cell to respond to external stimuli and put genes in the right place for expression. But I know this is quite a busy slide, but even with you can see here on the right-hand side, with the progeria cells, even when we take away the serum, no movement. And then the control panel shows the movement. So really nothing happens. And that's because of the lamin mutation. So what happens when you treat the cells with the FTIs, the drugs, and we're moving on to the next slide, you can restore this chromosome positioning and repositioning, should I say. So these chromosomes now can actively move. And hopefully you can see that in the graphical panel. But also there's some images showing that we also restore what is called nuclear myosin 1 beta. So it's a nuclear motor protein, and we think that it is involved in moving chromosomes around the nucleus. So if you knock it out, you lose the nuclear myosin, and then you can't move a chromosome. But in the HGPS cells, they have it disorganized. Okay, and on the side is a cartoon about how nuclear myosin might work attaching to DNA in the nucleus. So I'm moving on to the next slide. So we can restore not only the chromosome position, but the chromosome movement in the HGPS cells, the progeria cells, with the FTIs. Something we also wanted to look at was the shape of the nuclei. Do these drugs change the shape? And I think this is like one of those situations where you don't get the answer that you were expecting. It doesn't support your hypothesis, but it's also it's just as valid, because we found that we couldn't see any statistically significant change in the shape of the nuclei with all these different drug treatments. And I think that's really important, because we know that they're changing. They're restoring these amazing mechanisms inside the nuclei, but they're not changing the shape. Or the shapes are normally quite normal in these cells that are really aberrant. So that's a really important point to put home, that you need to select your assay, your endpoint for when you're looking at drugs. So we do have DNA damage foci, and we can see them with gamma H2AX. And this is another sort of cautionary tale, if you like. So when we treated with all the drugs and the combinations of drugs that were in this study, we found that zoldronic acid, when it was in the media, caused a lot of DNA damage. Okay, so we saw these gamma H2AX foci. However, when we did the same treatments and looked at these cells using COMET scans, so COMET assay, where broken DNA will travel further through the gel, we found that this DNA repair was not real. Okay, so it was something that had come up. We've got an expression of gamma H2AX, but when you actually physically look at the DNA and double strand breaks, they aren't there. So we could have gone to the children and told everybody that, or the families, that zoldronic acid is very dangerous, but actually we needed to do the assay better. So what came out of this as well, that rapamycin was very, very good at repairing the DNA. So that was good, but rapamycin, on the other hand, causes a lot of issues as well. So it has a problem with it has side effects. And so, but anyway, the children are now on, and I'll come to that, on rapamycin as well. FTI might also not be a great drug because it allows a buildup of toxic pre-lamin. Okay, so pre-lamin is where you have the farnesyl group still attached to the protein, and so that usually gets turned over very quickly. But when you use FTI, you get a buildup of the pre-lamin, which is just as toxic as the progerin. So that's not good either. Progerin is removed in drugs in cells that are treated then with FTIs and rapamycin. We do see that, so that's really important. So progerin is taken away, but pre-lamin is still there. But just to update you guys, this came through last night in the email, and these children are coming through the final stages of this drug treatment, and it's a combination of everolimus, which is basically rapamycin, combined with the FTI, which is called lonafarnib. So, and that's the end of the talk. So, thank you.
Thank you. Thanks, Professor Joe. It was a very nice talk. A lot of updates. So just so while working, I had the idea, testing the extracts and fraction on these Progeria cells. I did try sometime. But we want to make it work, which we want to collaborate and do. So while doing that, we're also planning some work on doing on DNA damage, as comment assay and gamma H2X is my favorite one.
Okay.
So I think
but
we want
maybe
to do that.
But maybe in collaboration, we've got the comets and the Metaphor system at Brunel, so we can. I mean, everybody uses it, Pravin. Everybody uses gamma H2AX. It's just a if you do belts and braces, as they say, if you look at both systems, then you're good to go.
Yeah, so while I was there, I was mentioning you about the fish and comment, the combining both the assays. So we can look DNA damage and the chromosome positioning in the same assay. So we can actually plan that. But from our side, we are more towards the natural products. So we have got here in Sahyadri experts in extracts. People are working on plants, so we have got some people who have got expertise in plant extracts, then fractions. Then we are also testing them on in vivo studies. So we got some expertise in that areas, and we want to have this collaboration so that way we can bring our expertise and your group expertise. Then Dr. Kill is there. He will be supervising us. So that's what we want to bring from this collaboration. In addition to that, we have got quite a few young people here, so we can actually go for some grant applications in Europe, UK, and in India. So that's what the other thing. And then the third thing is the co-innovation, which is part of Brunel. So last time we had the grant from them. so we can again go for the same thing. That's what we are actually planning from this collaboration. In addition to that, the larger MOU we can plan, in the future, as we, we'll discuss that. So that's from my side. I would like to now hand over to our principal sir, Dr. KV Otare sir, and he will have some updates and the ending notes.
Thank you, sir. Uh, Good afternoon, madam.
Good afternoon. Very nice to
Good
meet you.
afternoon. Nice to meet you. Thank you, Professor Joanna Bridger, ma'am, for your thoughts on Progeria research. We actually really enjoyed your conversation, and after your conversation, we are very eager to learn more about your research and what you are doing at your institute with your group. That is very important. Now, most of our students are at UG level, BPharm level. They are attending your webinar. So, to just highlight or to know our students what is a progeny of this disease or disorder, that just to remind them, I just mentioned here that our students may be seen in movie named as a Pa, right? It is casted by Amitabh Bachchan released in 2009. That is based on this particular disease. So they may remind that what is that particular disease is, and they may feel an interesting the research and area. Now, as this is a progressive genetic disorder, it is very critical to treat and handle. So, as an pharmacy students, pharmacy background, pharmacy peoples, I feel that there is a very big scope in the research. If we get a mentor like you for the research, then that will be more better for our faculty and students to be able to go in this area for the research. So, so as a Professor Joanna Bridger discussed with us today, there is a scope for research in this area, and as we are the pharmacists, we can take this opportunity to work on it. I thank you, madam, for your thoughts on Progeria research. We, the faculties of Nausayadri Institute of Pharmacy, our students, will definitely like to be associated with you for your research area. Now, in this line, today we are going for, I think, MOU, as Badesar has been discussed with you, for the research aspect. So, this MOU, basically, we are going to do with your group in Brunel University, and that MOU basically gives or provide us a new horizons in Nausayadri Institute of Pharmacy, and it will widen the scope of research in our institute. So that will be, we are very grateful for you, madam, for being agreeing to undergo this MOU. Thank you once again, madam, for your acceptance and giving guidelines, your thoughts, your creative ideas for our faculty and students. So it's very nice to get you as a speaker today on the occasion of this World Pharmacist Day. I thank you once again, Professor Joanna Bridger, for sparing your valuable time for us. And I'm also thankful to Dr. Badesar that he has tried a lot to bring you on this e-platform for us. Thank you once again, one and all. Thank you, ma'am.
Thank you very much.
Over to you, Badesar.
thanks everyone, and so we can now end the session. Thanks all. Thanks.
Thank you.